Hey, I'm going to take the mask off because it's kind of hard to hear me, but hopefully you will keep yours on. Now, what you see is the four horsemen on the left. Those are world-class problems. They've been in the Bible, they've been everything. Drought, malnutrition, disease, poverty, violence. Do we have a shot at any of those? Is that what biotech is about? I think it is. And so you look at those four horsemen on the right, they're much more the human condition, and you say, can we do this by taking core science, basic science, and really transforming it into solutions? Okay, I would like to look at this university in changing its name to the University of Global Problem Solving. But I think that's a step away. Let's just see if we can solve a few. Like today, let's go after drought. Now, drought is something that occurs in Montana about every 4 years, 3, 4, 5 years, sometimes 2 years in a row. And a farmer plants his crop and gets nothing. And in other parts of the world, like in Australia, they had 7 years of drought in a row. So, what's this course about? I think this course is that we give you some passion and we give you some vision. And we find the best people on campus for those kinds of things that are solving problems, maybe COVID type problems, maybe world food problems, but each has their own passion about it. And I've been serving as a coordinator of this course for a few years, and it seems to work. We don't tell you what to think. We don't expect you to take notes and write down all the stuff we say. We want you to develop your own passion. And if you do, by the end of the course, you should be able to write a 2-page essay on how you would spend $3 million to solve or go after a world-class problem. So, welcome to the club. That's what we're doing. We have a very good TA to help you. And, about face coverings. The MSU has set down some rules about mandatory masks and social distancing. We're all in this together, and let's keep it that way and keep all of us healthy. Shoot. There. Now I'm learning to work a computer. Okay, we created a new word. Why did we have this word? Because people think it just rains. But we think bacteria on the leaves of the plant, they are swept into the atmosphere and they do something we call bioprecipitation. And we've been working on this for a number of years, and it seems to be that it's a coarse science. These little things here are only a micron long and half a micron wide, and they're called bacteria. But the beauty of them is that they have 3,000 genes. They're problem solvers. They only produce gene products for 150 of those genes at any one time. So which ones? Can they predict the environment? What is their safety net? These are fantastic organisms, and the beauty of them is you can do an experiment, go home, do what you do, come back the next day, and the experiment can be done. So they're very fast. This guy, like E. coli, which is related to this one, they basically wrote the book for biochemistry. And without that, we wouldn't know what's going on in cells. We wouldn't know anything. And we're going to show you, I think, in this talk, about if you know what's going on in cells, you can really make a difference. These bacteria can be good guys or they can be bad guys. They can cause disease. They can produce vital nutrients you need for your body. And they can stretch your imagination. So about 50 years ago, I published this paper on the left. And basically it was computer science work that took 5 years, 200 tests on 200 different bacteria called Pseudomonas. All of them live on plants. But by not putting them on plants, and these bacteria, you can't tell them apart by looking at them, we found that the computer could sort them out according to their physiological activities, and those were how they determined what their host range was. So we call these bacteria Pseudomonas, short for Sue. Recently, we published a paper in PLOS about, 6 years ago, and we used DNA tests, and we could find these same groups of bacteria. But the difference is, with the physiological tests, we knew what they were doing. This was just a DNA fingerprint, and it didn't tell you much. So, I worked in a place called Connecticut after I got my doctorate at UC Berkeley, and I think I worked on a problem that was 300 acres. When I had a chance to come to Montana, where they have 4 or 5 million acres of wheat, I jumped at it. And they asked me because on a wet year, they'll get symptoms like this, and this is a barley plant. Looks like a wheat plant too. And it seems that they were losing about 5% yield in a wet year. So they brought me out and said, you're an expert at Pseudomonas, what are you going to do? So I said, well, I know how to kill these bacteria. I just take 2 tons, or actually 20 tons of a semi-load, and I treat it with a little compound called Bronopol, which is in shampoo, and voila, you can see the yellow bacteria here, but you can also see that we have a good zone of inhibition around Bronopol. So we treated 20 tons of seed, planted in 900 acres, and I felt good about that for about 3 days. On the third day, 3 weeks, and the farmer called me up and said, oh, that bacterium you're after, it's all over these fields. Could you fly back up here and figure out what went wrong? And so I took a Cessna up there, and I held petri dishes out the window, and sure enough, these bacteria that were in the field were also in the air. And as we approached the cloud, there were bacteria too. And when we got in the cloud, suddenly there were ice crystals hitting the petri dish. And wherever an ice crystal hit the dish, there was Pseudomonas. So we developed a theory that maybe these bacteria are doing something besides causing disease. These bacteria can grow, and this is an electron micrograph of a leaf surface, and you can see these bacteria can grow overnight, and they could go from 10 to 500 overnight, and then they fall off like dandruff. And sure enough, you can find them in the air. So they move around. Wouldn't you, if you were a bacterium, move around? You get tired of one place? So we developed a hypothesis, and we published it in 1982. Bacteria grow on plants. They're taken into the aerosols, and funny thing, they cause ice nucleation in rain, in the cloud, that glomps the water together, and we call that rainfall or snowfall. And we developed this idea in 1982, and nobody paid attention. But the idea behind bioprecipitation is that you take this weather, a precipitation highway coming off the Pacific coast, and as it goes across the continent, it can cause cycles of bioprecipitation. And maybe with the right crops and the bacteria, and the right bacteria, we can get even more rainfall out of this bioprecipitation, or this precipitation highway. And so we look at these bacteria. Are they special? And the answer is yes. Over 400 million years now, they've developed a method whereby they can freeze water at -2 degrees. Now, you all know that water freezes at 0, but that's not true. You need some kind of template, some kind of nucleation event. And in this case, they have a protein on their surface. But if you look at atmospheric dust, or forest aerosols, or pollen, or silver iodide that they dump out of airplanes, none of them compare to the effectiveness at -2 degrees. So these are the bacteria. They have a surface, a double membrane here, and on that membrane, if they're in the cold, they produce this little protein, a spring like that. And they only do it in the cold. So it's one of those 3,000 genes that only turns on under the right conditions. So here's a protein called the ice nucleation protein. And it has a DNA sequence in it, something like this. And there are 48 repeats of that. So the organism, the Pseudomonas, basically works like a Xerox machine in the gene, and it makes a whole lot of the same sequence there, and that lines up perfectly in a protein, causing water to form, or actually ice. And on the upper left you see Cindy Arcer. She was a student here, and then she went to UC Berkeley, and she studied these bacteria that cause frost, cause ice, and she is the first one to have cloned this gene. And you can see here the red letters wherever there was that particular sequence. And anyway, she's now a very famous scientist, probably had her picture on things like Newsweek magazine and things like that, and she came out of Montana. And this is Brent Christner. He likes to study glaciers, including in Antarctica. And he was wondering, well, is there a glacial record that we could find of these bacteria in good years and bad years, and could we correlate it? And then this is Boris Vinatzer. He's at Virginia Tech, and he loves to just clone these bacteria and figure out what all the genes are. And then the mastermind of all this is Cindy Morris, and she is in France right now, but she works on these bacteria too in the country of France. So if we look at this protein, we see that it has a crease right down the middle, and that's where the ice nucleation occurs. And this took a lot of work, but can we apply this? Can we use this in some way to create more rain? So let's look at that protein. It's basically like this pink part of a egg carton. And water, when it's bouncing around, it has a lot of energy in it. And when you freeze the water, and like these eggs, and you freeze it, they have to give off all that bouncing energy. And that's an interesting fact that occurs whenever you form ice, is it has to give off energy. And we'll see if we can give a little energy to this project now. What we did is found a piece of tin foil, and we placed it on an aluminum block, or floated it in super cold water, and we could drop raindrops on this, and wherever we dropped a raindrop, if there were ice nucleation bacteria in it, they froze. And that was really a quick and dirty way to find out if there were any raindrops out there and if they were forming from ice formation. And we think they are. And in fact, we know they are. We've looked at clouds, and as you go up in an airplane, as you know, it gets very cold. Airplanes usually fly up around here somewhere, minus 40 or 50. But at -2, these bacteria, they can cause ice to form. And I was working in Tunisia, which is in North Africa, for about a month, and we had clouds every day and no rain. And I was wondering, is that just the way the world is? Or is there something about our crops, is something that we're doing that may be able to be improved? So back to Montana. We look at these 4 major ski resorts that we have, and we gave a bottle of whiskey to each guy at the top of the lift and told him to collect snow all winter long using a sterilized shovel and putting it in an ice chest. At the end of the snow season, we melted that snow and we assayed it using that droplet technique, and we found that 70% of the snowfall that we get in Montana is precipitated by bacteria. Wow. Where do they come from? Do we have any idea? Well, NASA does, because NASA tracks storms. And this is a high-split method of tracking storms. And basically, storms started here, or here, or here, or here, and they end up in our ski resorts. And basically, we were able to say that we know where the bacteria are coming from, but we don't know much more than that. So the next question is, are all plants the same? Do they all produce these bacteria? And the answer is, these are in logs. Like 10 to the 3 is about 1,000, and 10 to the 6 would be about 1,000,000. So plants really differ. Then we looked at just a barley crop, and we found the same differing there. So depending on which barley you plant can have a great effect on how many bacteria can fly in the air from that field. So we then moved over to Syria, which is the International Center for Arid Research in Dry Areas, or Agriculture Research in Dry Areas. And we looked at all 100 varieties of wheat they have, and we found, again, the same thing. There's 1,000-fold difference in how many bacteria can be supported by a plant. And I think if you plant the green one, you're going to get more rain. That is our hypothesis. So we looked at all the bacteria on these leaves, and we found that one seemed to be really good. It didn't cause any disease except in pea plants, and they don't grow peas there. And it nucleates at -4 degrees, and it seems to be growing on the surface of the plant and doing something strange. Besides ice nucleation, it also protects the plant. It produces a natural fungicide that keeps that plant nice and clean and healthy for that bacterium. So we're starting to develop a plan. And the plan would be to find the right Pseudomonas, those that are strong ice nucleators, those can live on various plants and also protect the plant. And let's hope that we find them then in clouds and rain and snow, and hopefully they can make a difference. And can we put them on seed, and then they grow to the top of the plant and then get themselves on seed for the next year? And so our bioprecipitation is starting to have traction. Meteorologists are looking at it now. And it seems to me that at least in some parts of the world, it may work. So if you look, like, you got to really get busy here and look for 40 days on wheat plants or barley plants and see if the bacteria, 10 bacteria, can go up to 1,000,000 bacteria in 40 days, and they can. And so that suggests that our plants can be a launch pad. We can find the right plants with the right bacterium, and voila. Maybe we have a launch pad, and maybe that launch pad is just a field that can produce 10 to the 12 INA bacteria every day. And now that could be a game changer. And as you can see, it starts with some basic science. It's still a dream, but we think we're making headway. We think we're understanding where to go, where the drought is worst. This was done by NASA, and all these brown areas are in fact where they had 5 years of drought. And Texas, they've had drought. California, they've had drought. Australia, they had 7 years of bad drought. So maybe we're doing something wrong. Maybe we can, by maybe less grazing of animals over the green leaves that produce our bacteria, maybe we can do something a little bit better and not take rainfall for just what's given. This Keith Big in Australia was very instrumental because he's been trying to cause rain for 50 years. And what he found is that when they did get a fair amount of rain, suddenly they got more rain in 6 days later. And he can only explain that by biology. So we have a model, and we tried it out on the western United States. And these blue areas are where we thought we were getting actually more rainfall due to bioprecipitation. And if we focus down, we see that the Central Valley of California could be really critical. That's where there are people fighting over water. The almond growers, the big cities like Los Angeles and San Francisco, they're all fighting for the same amount of water, kind of like a zero-sum game. And we think we can change that by planting the right crops with ice nucleation ability bacteria right here in the Central Valley and harvesting this precipitation highway that flows across the Cascades, across the Central Valley, and all the way to the Sierras. Now, most of the rivers from the Sierras flow back to the west, and they're used to irrigate or to feed people in the cities, but whatever, they flow back. And so you can have a giant washing machine with maybe ice nucleation bacteria being very much a part of it. So the general model, and we presented this at the Paris climate change meeting, and a lot of people are referring to this paper. And basically what it says is that you get the bacteria that go up. This is the precipitation highway. They cause rain, which causes more growth and more plants and more rains and more cycles all the way across the continent. Now, one thing, if you look at a Google Earth, you'll see that there's a lake here, and that's where it's coolest. But the yellow places are where you have freshly plowed land with no plants. When you get some plants, you get cooler. And in fact, we published the fact that a tree, just a normal tree like that one out there, can cool the atmosphere as much as an air conditioner, which uses a lot of electricity. So, what do? We do biotech. We take some core scientists who understand what bacteria are and what proteins are and so on, and then we figure out how maybe they can solve bacteria bioprecipitation problems, and we make sure that they're safe, they're good nucleators. We can mark them and track them so that if we do something wrong, we're held to blame. And we can coat seed with them, plant the seed, follow them down, and see if we can do anything with rainfall. And if we can't, if we fail, we can hire some NASA people who will try to explain why a launch failed. To be successful requires a bunch of different kinds of scientists and innovators, and that's what we have going on here at Montana State. So, there are your world-class problems. That's a vacant lot for you. If we can convince you to be part of a team doing biotechnology, maybe we can change the name of the university. So, I like to dabble in poetry. When the sun goes down and the various birds streak across the sky, destination bound for some safe haven, and Venus pokes her face through the twilight, I try to take some time to see if my effort made a difference, not compliant with the zero sum. I want intellectual progeny, a finer kind of question, important for a finer kind of answer, not diluted by generation, not reduced to consensus trends of the day, but true brilliance, outshining Venus herself, emerging from this cold and windswept valley. And these are some of the students that have launched out of our lab, and they're doing great things in the world, and we expect more. Thank you.